Okay. Is that my computer? Oh, get feedback? No. Okay. Yes, I do.
Yeah, I think that's like.
Hold on.
That's the stuff they got back. Like, I think it was,
There we go. All right, I got it now. Okay, sorry. So anyway, thank you everybody for joining us for seminar this week. Today we have a speaker, Ashlyn Smith. For those of you on Zoom and in person that are registered for the seminar, I'm going to, for the in-person folks, I'm going to send around a sign-up sheet, just initial next to your name under the first date that's there. For the folks on Zoom, we will note your presence. Just make sure that the name that's showing up on your screen is the one that you're registered for the seminar under. And if it's not, let us know so we can document your presence. So without further ado, Dr. Deborah Miller is here to introduce Ashlyn. And so Dr. Miller, I'm going to hand it right over to you.
Thank you. Hi, everybody. I'm excited to tell you a little bit about Ashlyn and let her take it from there. Ashlyn got her bachelor's degree from the University of Central Florida in interdisciplinary studies with a minor in environmental science. She then moved back up here to the Panhandle so that she could work for the Florida DEP. In the ecosystem restoration science section and through the environmental resource permitting section. She then came back to UF and worked as a lab technician and began her graduate work, and then went to work for the Atlanta Botanical Garden as the Gulf Coast coordinator for 4 years. So she was there quite a while. She came back to UF. We were able to court her to come back as a project manager for a very large-scale dune restoration project that we're involved with. Graduated and has now got a fellowship with the National Academy of Sciences, very prestigious fellowship. And she's working in Mississippi-based Restore Act Center of Excellence. So take it away, Ashlyn.
Hi, everybody. Thanks, Dr. Miller. I'm really excited to talk to you all today about my PhD research. I'm a very recent graduate from SNRE. I graduated this summer, and yeah, so my research is about the different restoration techniques and approaches for wet prairies in the Florida Panhandle, and how a few novel approaches that we utilized may lead to improved restoration outcomes. Oh, can I progress? Let's see. Oh, okay. Oh, how do I go back? There we go. Sorry about that. So as the title implies, I was working in wet prairies. These are herbaceous ecosystems, and they are in need of restoration. And the primary reason why they need restoration is due to shrub encroachment. Shrub encroachment in herbaceous ecosystems is a global issue, and it's defined as an increased density of woody plants, bushes, and shrubs, of course. They can either be native or not native in systems that are naturally herbaceous. You can see the diagram towards the bottom of the screen, shifting from an ecosystem with a high number of plant species in the ground layer to eventually only being a few species in the sub-canopy and canopy level. And those species are now woody. Shrub encroachment does lead to soil carbon and nitrogen increases. And for the ecosystem that I've been working in, these wet prairies in the Florida Panhandle, these are naturally nutrient poor ecosystems. So increases in soil carbon nitrogen are negative. And of course, there are reductions in light, subsequently herbaceous vegetation and plant species richness as well with shrub encroachment. So the wet prairies that I was working in, they are terrestrial wet prairies located in the Northwest Florida Panhandle in Walton County, Florida. And everyone, wants to emphasize the importance and the uniqueness of their ecosystem. And I really do feel like these wet prairies, they're an excellent system to work in, and they are pretty unique. They have very high plant diversity as well as plant endemism, meaning that there are species that occur in these wet prairies that only occur in this area of the world, and they occur nowhere else. And to sort of highlight that and their importance, there's a quote at the bottom from Folkert from 1982, stating that well over half of the North American insectivorous species, so by insectivorous species, I mean those pitcher plants and the sundews, these species occur along the Gulf Coast, with as many as 13 species in 4 genera occurring in a single wet prairie. And then the image on the right is something else unique to Walton County, Florida, and those are coastal dune lakes. They are primarily freshwater lakes, but they do have intermittent connectivity with a saltwater source. And of course, we're in the Florida Panhandle, so that saltwater source is the Gulf of Mexico. Now you might be asking yourself, why are you talking to me about these terrestrial wet prairies and coastal dune lakes on the same slide? Well, it just so happens that the wet prairies that this restoration work occurred in are immediately adjacent to the freshwater streams that flow into these coastal dune lakes and then have that intermittent connectivity with the Gulf of Mexico. And this is made more clear by this aerial imagery. So the image on the left is from 1941, and the red line on the image is just a reference line. But the emphasis I wanted to put on this slide was that the yellow polygons are the historic herbaceous wet prairie ecosystems. The darker linear signatures next to those are the freshwater streams that flow into those coastal dune lakes. And here you can see Deer Lake and Camp Creek Lake are just 2 examples. So all this restoration work occurred in the watersheds of these 2 coastal dune lakes. And the image on the right, by 2014, that herbaceous ecosystem has been completely closed in and shaded out by a canopy layer and has experienced shrub encroachment. The red line is just a reference line. It's the same road. I just wanted to give a reference point. The primary culprit for this shrub encroachment is black tie or Cliftonia monophylla. It is a native shrub. I did want to emphasize that. It's native, but it is encroaching out of its natural boundary, and it should occur immediately adjacent to those freshwater streams, but it encroaches outside of those bounds and into the herbaceous ecosystems. But its natural range is pretty restricted to the lower portions of the southeast. And this is what that looks like on the ground. So the image on the left is a wet prairie and what it likely looked like in its historic state. And the image on the right is a shrub bog. Shrubs have completely encroached into this ecosystem. Oftentimes, the only way we know that this shrub bog once was a wet prairie was through historic aerial imagery, or there's remnant herbaceous vegetation and wet prairie vegetation at the ground layer inside of some of these shrub bogs. So before approaching this project with any restoration action or technique, I first like to look into a little bit of ecological theory before, just bear with me, before going into the restoration approach. So many of you in the room are probably familiar with Holling's stable state theory and using this sort of ball and cup diagram to explain the stable states. So the wet prairie on the left is one stable state. The shrub bog on the right is the next stable state. And imagine this ball here is the ecosystem itself. And this is my fancy animation skills at work here. So as the ecosystem is being encroached by shrubs, it's moving up this hill, right? But in the southeast, we have this natural disturbance, this natural historic disturbance that comes through and maintains this herbaceous vegetation, and that is prescribed fire. So yes, this system is maintained in the wet prairie state with a natural disturbance, prescribed fire in the southeast. Now, when fire is removed from that system, the ecosystem can freely be encroached by shrubs. And after crossing this critical threshold, it then easily moves into its new stable state, where we know based on this theory and based on previous research, it's now going to be extremely difficult to return this system back to its historic wet prairie state. And oftentimes that original disturbance, so fire, is not going to be enough to get it back over that hump and into this other stable state. So throughout the presentation, I'll be referring to this wet prairie condition as the reference condition and the shrub bog ecosystem as the degraded condition or the control condition. I'll be talking to you about 3 components of my PhD research. We did conduct, prior to any restoration action, we conducted a seed bank experiment where we wanted to know the restoration potential from the soil seed bank, as well as the soil seed bank could provide us with some guidance as to future restoration action. And of course, then we apply restoration treatments, monitor the response of ground cover vegetation to those restoration treatments. And simultaneously, we analyze soil, physical, and chemical properties in association with the restoration treatments, as well as through time along the restoration trajectory. So first, I'll talk to you all about the seed bank study. This was the seed bank project design. Soil seed bank samples were collected from 4 reference wet prairies and 6 degraded wet prairies. Within those prairies, samples were collected from 3 slope positions at 2 sample depths, as well as 2 stratification levels. So samples were split into either being cold stratified at 4 degrees Celsius for 40 days, or they were not cold stratified. Samples were then placed in a randomized complete block design across 10 greenhouse blocks in the greenhouse for a total of 1,200 samples. An additional component was to compare what was emerging from the soil seed bank to what was occurring in the extant vegetation at reference and degraded wet prairies. And the greenhouse study duration was 10 months. This is a map showing you where those degraded and reference wet prairies are located for this study. The circles are the degraded wet prairies and the diamonds are the reference wet prairies. And you could see at the bottom the coastal dune lakes in there as well. So Deer Lake and then Camp Creek Lake on the right. But the main point of this slide was to show you that these reference wet prairies that we used as the target and the goal for this restoration action are not far away from the degraded wet prairies. They occur along the same soil series. They're within the same watershed boundaries. And yeah, so we're not using a reference condition, miles and miles away or across counties or anything like that. These are the soil samples being separated in the lab, and the image on the right is showing the greenhouse experiment design. Now, this was a lot of data analysis has been done, but what I'm going to do today for this presentation is on the comparison between what was emerging from the soil seed bank compared to the extant vegetation at the reference and degraded sites. So to analyze that data, the response variable was species composition, or the presence-absence of all species, was simultaneously analyzed based on different groups using a permutational analysis of variance. And this takes this multidimensional data and reduces it down to 2 axes, visualized with non-metric multidimensional scaling. But essentially, it just allows for the statistical comparison between groups looking at multiple species at once. My grouping was based on condition. So either it was reference prairie or degraded prairie and its location. So it was either on-site, extant, or it emerged from the soil seed bank. So the non-metric multidimensional scaling allows us to visualize this data in two-dimensional space. And the data points, the individual data points are all the data points that went into the analysis and the visualization. The circles are showing where the concentration of those data points are located, and the colors are based on that condition location grouping. So, not surprising, right? The extant vegetation of degraded wet prairies and reference wet prairies is statistically significantly different from each other. That's based on the Permanova. Excuse me. Also, the extant vegetation from both reference and degraded wet prairies was not similar to what was emerging from the soil seed bank. But I think most importantly, this analysis result showed us that what's occurring in the degraded soil seed bank from the degraded wet prairies is not at all similar to what's occurring in the reference wet prairies on site. So this tells us that, number one, we can't really necessarily, we're not going to be able to rely on the soil seed bank to revegetate this area after restoration action. It also let us know that, okay, if we want to take some more novel or maybe more aggressive restoration approaches, we're not going to be harming the soil seed bank and setting us back in the negative direction on our restoration trajectory. So now I'll get into the vegetation and the response of the ground cover vegetation to the restoration treatments. First, I need to tell you about what the restoration treatments were. So there were 4 main broad level approaches. Clearing, which is always going to be designated with a C throughout the presentation. And this is the mechanical removal of the woody vegetation. Scraping, represented by an S, was the mechanical removal of the organic matter that has built up in these degraded wet prairies over decades and decades of shrub encroachment. And the organic material was scraped away down to where bare mineral soil was visible. Diaspore means the transfer of diaspore material from the reference wet prairies onto the restoration treatment areas. So diaspore material collection just means we didn't do a targeted seed collection. We didn't target any particular species or their fruit. We collected all above ground plant material and transferred it, and it looks, sort of like a hay onto these restoration areas. We were required to do this by hand. So it was the only non-mechanized portion of this. And then the treatment on the right is, of course, fire and prescribed, designated with a B. So all 4 of these treatments and all combination of these treatments were applied in a randomized complete block design with a split restriction on the randomization across 8 sites, 8 degraded sites in Deer Lake State Park. Of course, we also had the 4 reference wet prairies that we were monitoring the vegetation alongside of these restoration areas. And these treatments were also applied at the upper, middle, and bottom slope positions. And this diagram does look a little bit complicated, but I'm going to break it down a little bit. And here, again, just showing you it's the same degraded wet prairies, same reference wet prairies. We just added 2 additional degraded wet prairies for the restoration treatments. The complete list of restoration treatments is on the right, and that experimental design schematic is on the left. So the way the split plot design works is, let's say we're talking about the control condition. We're not doing anything restoration treatment wise. So the first split is either, was it cleared and removed of woody shrubs mechanically, or was it not? So this is the control, and it was not. The next split would be not scraped, And then of course there was diaspora material added and it did not receive prescribed fire. So that's maybe, this is an example of how maybe one of the sites was laid out. And then this is what the control looked like on the ground. The cleared plus scraped. This time we're going to go on the cleared split side of the design, then the scraped. However, there was no diaspora material or prescribed fire. And that's what this looked like field. So all of the area image was, but the larger rectangle where you can see the bare mineral soil clearly exposed was scraped away of the organic material buildup. But for this treatment, there was no diaspora application or prescribed fire. And then you can see the upper, middle, and bottom slope positions there as well. And then the cleared plus diaspora plus burned, again, we're on the cleared side, did receive diaspora application. However, it was not scraped, but it did have prescribed fire applied. And that's what this one looked like. So it's cleared, not scraped, did have diaspora, and was burned. And we also did diaspora alone, of course. So not cleared, did receive diaspora, was not scraped, and did not have prescribed fire applied. That's what that looked like in the field. You might have noticed some asterisks next to a few of these treatments. I've highlighted them there in yellow now. So this is field work, right? Things happen and some things get burned that maybe shouldn't have gotten burned. And there are other things that maybe should have gotten burned that didn't. That's field work, that's the nature of it. But these treatments, we didn't have enough replication to include them in the data analysis. So they were removed prior to data analysis. Now I'll get into the results. The final list of restoration treatments that were used in the analysis is on the left. And we did conduct seasonal sampling in the spring, summer, and fall for 2 years. So it's a total of about 200 sampling quadrats. And the percent cover of every species was recorded in each of those 1 meter square quadrats. So to analyze this vegetation data, the response variables were percent cover functional groups. So I decided to use functional groups here because a lot of times the land managers will do this because they don't necessarily have the time or the resources or the staff with the skills to identify plant species, plants down to the species level. So they might have a goal, for example, of they want to reduce woody cover by 50%. And so they're able to quickly assess that. And so breaking the data down this way and analyzing it this way makes it more meaningful for them. But of course, I also looked at species richness, which is telling us the number of species occurring in these restoration treatments, as well as species composition. So just because a treatment may have more species, it doesn't necessarily mean that it's the same suite of species occurring at the reference site or, our end goal, right? So the methods for the percent cover of functional groups and species richness was analyzed with generalized linear mix models. And then again, for species composition, I used permutational analysis of variance. So for the functional groups, for the woody cover, there were 3 treatments that had similar woody cover to reference prairies. And when I say similar to, I mean not significantly different from the reference. So the goal is to be like the reference and not significantly different from it. And these 3 treatments did do that as far as the woody cover went. So they were cleared plus scraped plus burned, cleared plus scraped plus diaspora plus burned treatments. All had similar woody cover to reference prairies. For the graminoids, there were no treatments that produced similar cover to reference prairies. So these reference prairies are very high in their graminoid cover. 80 to 100% is very common. Sometimes if there's overlapping structure with the graminoid cover, it's above 100%. So none of the restoration treatments have quite gotten to that reference prairie level as far as graminoid cover goes. And for forbs, there were two treatments that had similar forb cover to reference prairies, and those were the cleared plus scrape plus diaspore and the cleared plus scrape plus diaspore plus burn treatments. Now, let's say I'm a land manager and I want to get the most bang for my buck, right? I want to know if my only goal is to reduce woody cover, I'm not really concerned about anything else. What's the bare minimum I need to do? Well, the least common denominator here is that you need to clear away the woody vegetation as well as scraping away that accumulation of organic material. However, if you are concerned with the forbs, then you want to clear and scrape and bring in diaspore material from another location. And here's what that looks like in the field. So the yellow box is showing the area that was scraped. The entire picture was cleared at the same time, but the area in yellow was scraped, the organic material was scraped away from that. And just to give you a little bit more context, the shrub to the left of the picture is well overhead. It was taken about 3 years post-restoration. And here's an example of what the clearing and scraping and diaspore transfer looked like in the field. So in the background, that area was not scraped. The foreground was scraped. The white quadrat is showing what the cleared plus scraped looks like without the diaspore material being brought in. And then the addition of diaspore material, from this photo clearly showing an increase in the forb cover. So for species richness, like graminoid cover, there were no treatments with similar species richness to the reference wet prairies. And that's because these reference wet prairies are extremely high in the number of species that occur in them. So the average was 20 species per meter square, and none of the restoration treatments yet have reached that level of species richness quite yet. For composition, again, this is similar to those the visualization for the seed bank results, right? Now we've got all the data on 2 axes, and all of the points are representing the data points that went into the analysis. The colors represent the restoration treatments. The circles are showing where that particular treatment, where those data points are concentrated. And the permanent results for species composition were like that of the graminoid cover and the species richness. So every restoration treatment was significantly different in species composition from the reference condition. But visualizing the data this way along these 2 axes does show that the restoration treatment that gets us the closest in species composition to that reference condition is the cleared plus scraped plus diaspore plus burned treatment in red. The text around the outsides of the figure, those are individual species, and the direction and the distance from the center that those species are indicates the pull, essentially the pull that they have in separating the different restoration treatments from one another. So the species on the left are all woody species, and the species on the right are those herbaceous species, and you can see those are much more close in proximity to that reference condition. And I thought you all might want to see some pictures of some of these plants. So you already know about Cliftonia monophylla. Again, it's on the left side. And Lyonia lucida is another woody species over there on the left. So some of these herbaceous species that are contributing to the pull of that reference condition to the bottom right are Lobelia brevifolia, Baldwina uniflora, Carphephorus pseudoliaturus, Polygola lutea, Rexia alphinus, Coreopsis floridana, Liaturus spicata, and the Xyris stricta. So to summarize all this, like I said, if my goal is to just reduce the woody cover, the bare minimum I need to do is clear and scrape. However, if I do want to increase the cover of forbs, maybe I'm worried about the pollinator species in the area, I would want to also bring in diaspore material. However, if I want the most similar species composition to that of the reference prairie condition, then I would clear, scrape, diaspore, apply the diaspore material, and burn it. All right, now I'm going to talk about the results of the soil physical and chemical properties associated with the different restoration treatments. So this is that restoration treatment design. Again, the purple circles are showing the locations, the specific treatments where these soil samples were collected. They were only collected from a subset of restoration treatments and only from the upper and bottom slope positions. And again, there were 8 restoration sites, and we had 4 reference wet prairies where we also collected soil samples from for comparison. So the restoration treatments, the final list for the soil analysis are listed there on the left. They were control, so doing nothing, clearing, scraping, clearing plus scraping, clearing and burning, and clearing, scraping and burning, and then of course the reference condition. Soil samples were collected annually in 2022, 2023, and 2024. The 2022 sampling event was immediately following restoration action. 2023 was 1 year post, and 2024 was 2 years post. There was a total of 74 soil samples. Then of course we had to collect double that for the bulk density sample, which was a total of 222 rows of data. Every soil sample was analyzed for this list of soil variables on the right. And again, us, ecologists, we like to look at everything simultaneously and just help gauge what's going on. So the method here is a little bit different. I used a canonical discriminant analysis. Some of you may be more familiar with a principal component analysis. That's sort of what they call a free ordination or free analysis. The canonical discriminant allowed me to tell the model, I've already restoration tree have these predetermined groups, and it was distinguishing the difference between the groups instead of just a free ordination. That's the difference. So again, all of the data is reduced down to 2 primary dimensions. And here it's called canonical axis 1, which is the x-axis, and canonical axis 2, which is the y-axis. This analysis also tells us the percent of the variation explained by that particular axis. So here, 50% of the variation and the difference between these groups was explained just along the x-axis alone. An additional 20% of the variation in the data was explained along the y-axis. So the shapes here are each of the restoration treatments. Instead of displaying all the data points in one figure, these points represent the data centroid values. And I'm going to go over, and again, that text and the arrows are showing which soil variables are contributing to the pool and the difference in the data in that particular direction. But I am going to break this down a little bit further. So here, this is the reference centroid value circled in red. So if we're looking just at the x-axis only, the cleared plus burned and the cleared treatments along the x-axis are most similar to the reference prairie condition. However, if we're looking at the y-axis, then the cleared plus scraped and the cleared plus scrape plus burn treatments are most similar to that reference prairie condition. So again, let's go through examples. So let's say we're looking at bulk density. This figure tells us that the data points, as they move from right to left along the x-axis, the mean bulk density values for that treatment should be increasing. So these are the mean bulk density values for the different restoration treatments. So the star, the cleared treatment, and the cross, the cleared plus burn treatment, line up along that x-axis, right? And they have the same mean bulk density values as the reference condition. But if we look at pH, which has a little bit more pull along the y-axis, the mean pH values for the cleared and scraped and the cleared and scraped and burn treatments are, the mean values are the same as the reference condition. And then if we pick a soil variable on the right, so potassium, the mean potassium values now moving from right to left are going to decrease. So the control treatment and the scraped only treatment having higher mean potassium values, and then it's lowering as you move left along the x-axis. So to summarize the soil results, the cleared plus scraped and the cleared plus scraped plus burn treatments are the most dissimilar from the soil parameters in that control or degraded wet prairie condition. However, the cleared and the cleared plus burn treatments are the most similar to the reference wet prairie condition. So this is saying that the scraping away of that organic material is reducing the organic matter, it's reducing the soil nutrients, soil nutrients contained in the soil to those beyond that reference condition. And this is not super surprising because there have been others who've utilized this scraping technique in wet prairies in Europe, and they found a similar result, that it scrapes it away and it does bring conditions beyond that of what are at the reference condition. And based on those vegetation results, it doesn't seem to be hindering the establishment of the reference prairie vegetation on the restoration sites. So the big takeaway, right, is that traditional restoration techniques in combination with some of these new novel approaches are going to lead to improved restoration outcomes and faster ecosystem recovery following decades of shrub encroachment. However, there's a caveat there, right? When these novel approaches are appropriate. So for example, you don't want to scrape away the root masses and organic matter built from a wet prairie, from a degraded wet prairie, deep slope, right? Because then you might have some erosion issues. It may also not be appropriate to collect diaspore material in such a broad capacity and transfer it to the restoration area. If your reference wet prairie is small or isolated, you wouldn't want to take away plant material or seed sources from that reference wet prairie. And of course, I need to thank the partners involved with this work. The University of Florida did partner with the Atlanta Botanical Garden. We were funded by the US EPA. The results and the views and opinions expressed by me during this presentation do not reflect the United States Environmental Protection Agency. And I also need to thank the Florida Park Service and the Florida Forest Service for allowing us access to these sites and for conducting these experimental restoration treatments within the bounds of the lands that they manage. So thanks to them as well. And with that, I will take any questions.
Hello. Great presentation. I was just curious if you might be able to go back to the slide where you took all of the soil variables.
Sure.
I was just wondering, the extraction process that you did for phosphorus, like, was it like a Malic 3 extraction?
Yes, because they are very acidic.
Okay.
Mm-hmm.
And then how did you measure, cation exchange capacity?
So, these soil samples were sent to the Waters Laboratory in Camilla, Georgia. So I did not do it myself. And the detailed math are in my dissertation. I don't remember off the top of my head.
Sure. Yeah, no worries. I'd love to read the dissertation. I'm doing a lot of work with soil carbon and soil phosphorus. So, learning from somebody who's already finished it, anything that I may have missed, I think would be great. But overall, really, really cool. I love the figures. I love that figure, including all of your soil data in one. That was beautiful. Very nicely done. Thank you.
Thanks, Bruno. Yeah, feel free to email me. Yeah, and if any of you are like introverts, you don't feel comfortable asking questions right now, email me. I'm so happy to talk about this. Obviously, I'm really excited about it, and yeah, happy to chat.
Thanks again.
Just put my email back up. Joe?
Is that? Hello.
Yes, thank you for the presentation. How long after, how long after all your CS and D did you wait to burn?
Yeah, that's a great question. I should have talked about that. Sorry. It was about 4 months. So the the diaspore material was applied in November, and then we burned in March. And
Thank you.
it surprised me, right, that the burning actually appears to have helped because all the diaspore material was burnt up, but it didn't seem to affect the ability of those plants to get established.
Are you going to continue burning and monitoring?
We have one more monitoring event scheduled for October, and then from there it's up to the park.
You think?
Hello.
Hi.
Your presentation was so beautiful and you did such a nice job presenting it. I learned a lot, but I wanted to ask, what did you do with all the material that was scraped?
Yeah, that's a good question as well. It was sprinkled into the uplands and then consumed during regular prescribed fire for the park and the state forest. Yeah, and that is something that would need to be looked into a bit further, right? If this is going to be expanded into larger areas, because that was a concern for the park. And the state forest, and rightfully so. But we were able to manage the relatively small amounts just with regular prescribed fire and spreading it out into the uplands.
Okay, thank you.
Welcome.
Any other questions?
Hi, regarding your slide with like the before and after of like I think it was like 1914 to 1941 or something. How much do you think like urbanization played a part in like the degradation of the grasslands?
Oh yeah, a lot. I didn't get it right, but with increases in coastal development, these land managers are having a really hard time applying prescribed fire. But there was also that point in time where people were moving into the area and fire was seen as negative, right? So it was completely removed from the ecosystem. And then now that they're trying to reinstate it, it's that much more difficult because there's always a structure around and some sort of consideration to account for. This particular site is located between Highway 98 and Highway 30A, Alton County, and those are both very touristy areas. So yeah, it does have a lot to do with the degradation of the system for sure. Does that answer your question?
Yeah, for sure. I had one more. I know that you refer to the bog as like the degraded site, but like what are like the ecological consequences of like moving from the grassland to like the bog?
Right, so those increases in soil carbon and nitrogen that I mentioned are associated with shrub encroachment. It's just not the natural state for the ecosystem. There's also a concern that because these wet prairies are located right next to those freshwater streams that are then flowing into these super rare coastal dune lakes, that we could possibly be increasing the nutrients going into the coastal dune lakes as well. And then just overall lowering of biodiversity.
Thank you.
Other questions in person? Or online? I'm not seeing anything in the chat. Okay. Great. All right. Well, thank you very much, everybody. Ashlyn, thank you so much for joining us today and sharing your work with us. That was really interesting. And Dr. Miller, I can't see if you're still here, but if you are, thank you very much for doing the introduction. For those of you, well, pretty much anybody, but like, for those of you who are registered for the seminar, if you have any questions for me or any attendance issues, let me know. If anybody has any questions in general, also feel free to get in touch with me anytime. Otherwise, I will see you all next week. Thank you.
Thank you, guys. Bye.